A very warm good evening to one and all. First of all, I am taking this opportunity to thank the God Almighty for His eternal blessings that have been showered on all of us. Nothing would have been possible without His blessings. A warm welcome to those who join me here from the entire Department of Botany in collaboration with Department of Zoology and IQAC HPR College, Bangalore. I wholeheartedly invite you to the national webinar on the topic, the impact of structural vaccinology and bioinformatics on vaccine design and development. On behalf of the entire crew, I invite the Honorable Resource Person Dr. Mohd Akif, Assistant Professor, Department of Biochemistry, University of Hyderabad. Sir, we are so glad to have your esteemed presence here. Your words would surely be enshrined in our hearts. Now, I invite Dr. K. Jaykumar, Head of the Department of Botany, to introduce and welcome the resource person. Welcome, sir.
Thank you, Anjali. Good afternoon, and welcome back to the second and the last day of our national webinar series. I would like to brief yesterday's session. We had a talk by Dr. Irfan Ahmad Ghazi, Assistant Professor from University of Hyderabad. He reflected on basics and applications of functional genomics, structural genomics, and gene mining, with a special emphasis to disease-resistant rice genome development. And today, our second talk is going to be handled by Dr. Mohd Akif from University of Hyderabad on the impact of structural vaccinology and bioinformatics on vaccine design and development. So let me introduce our chief guest, Dr. Mohd Akif. Currently, he is working as assistant professor in Department of Biochemistry, School of Life Science, University of Hyderabad. He also served as postdoctoral research officer in University of Bath, United Kingdom, and as a research associate in Center of DNA Fingerprinting and Diagnostics, Hyderabad. Academically, he has an excellent track record. I would mention a few of them here. He completed his PhD in structural biology from Center for DNA Fingerprinting and Diagnostic, Hyderabad in 2008. He received a Raman Fellowship in 2014. He received a Young Scientist Travel Fellowship in 2006 and many more. He has more than 20 publications in high quality national and international journals. Sir, we are extremely glad to have you as our chair resource person for today's program. I welcome you, sir, on behalf of my own self, on behalf of Department of Botany, Department of Zoology and IQC, and on behalf of all the participants gathered here. Thank you.
Thank you very much. Thank you very much for kind introduction. And let me share my presentation. Is my presentation visible for all of you?
Yeah, it's visible.
Yeah. It's really honor. I'm really thankful to organizing committee who has given me chance to speak about my research and some of this importance of structural vaccinology. And I'm, as I was discussing Dr. Pillai, that I would be odd man out because I know this organizing committee mostly probably from botany department. But anyway, but she mentioned that the other participants from across the different discipline also. I'm really happy that they are really interested to listen my talk. Once again, I will really thankful to, for all of you for giving the opportunity. The title of my topic is like impact of structural vaccinology and bioinformatics on vaccine design and development. Let me briefly explain about what is the structural vaccinology and bioinformatics and vaccine. The first, I will pick up the vaccine. As everyone knows, the vaccine is very important. These are all of us know about what the vaccine. Really, we are rushing around to get the coronavirus vaccine. The vaccine is basically the biological preparation that provides the acquired immunity or the particular infectious diseases. The vaccine could be the killed viruses, kills microbe, toxins, or surface protein. And if you recall the background of vaccine, Edward Jenner is the first person who discovered vaccine in 18th century. uh, for cowpox and then smallpox. Later on, 19th century is basically dominated by the vaccines against rabies, cholera, and then typhoid. And 20th century was basically uh, um, uh, uh, dominated by diphtheria virus, tetanus, and then polio. And this century, if you see, the vaccine is continue to play high significant, but we are keep on expanding our role, its role, and then eliminating a disease such as many respiratory viruses, human HIV also, and then more recently that malaria, tuberculosis, and Ebola and COVID-19, they are the target for better vaccine design. When I say vaccine design, which mention the vaccine which basically induce your immune system. Everyone has an immune system, robust immune system. Any pathogen which comes under the body starts trying to kill the bacteria or viruses through its own immune system. And then we say the body's defense against infectious diseases is called immune system. And this has basically two types: innate immunity and adaptive immunity. Innate immunity, which is the first line of defense, any infection which comes through our body through the rapid non-specific responses are triggered and through the innate immunity. And the most of the recognition of the conserved structure present in the many microorganisms was basically the trigger point for innate immunity. But we have adaptive immune response also, which is a second line of defense, which is tailored to an individual threat. That means it can be tailored. It is very specific to the specific agent. And in this case, most of the time we develop a memory cell which persists and enable more rapid and potent response against the infection. So there's a basic background what we have what immune system does against any infectious organism. And slightly elaborate, but this adaptive immune response is the two arms. One is called humoral arm, second is the cell-mediated immunity. Humoral arm is basically the antibody mediated, which we see the antibody is raised in your body because of infection. That's more than sufficient to cope up these pathogen. And same time, the second arm, which is called cellular immunity response, which is a T cell response, also activated, and which triggers basically the memory cell and all. Usually, many of this antigen are in form of whole organism, in form of lipopolysaccharide, in form of proteins. Any agent which comes into the body, it has to be phagocytized by antigen presenting cells, and these antigen presenting cells basically chop off this molecule and present to the MHC Class-1, and basically this slide shows how the antigen basically is presented to the T cell to trigger for your memory cell and other immune response. Now, these are the basic background. Now, what exactly happening here? When we talk about the vaccines have been prepared in a. This is one of the slides which I found is it has been classified. If you talk about the vaccinology, has three different stages or three different methods. One is called conventional vaccinology, another is a reverse vaccinology, and the way near right now what we are in era of structural vaccinology. The conventional vaccinology is basically means that you take the organism itself, make it attenuated, and these microorganisms are is administered into the body to activate the immune system. This is all conventional vaccinology, and then it takes almost 5 to 15 years to develop a vaccine against any pathogen using the conventional vaccinology. But if you talk about last 1 decade or 2 decades, we have a plenty of genome available. If you talk about any organism, if you talk about any pathogen, whose genome data is available. Like basically last 2 decades, we have a plenty of genome information. Hence, that the other approach, which is very prevalent last 2 decades, in like 2000 to 2005, or in fact, even till now also, is called reverse vaccinology. What do you mean by reverse vaccinology? Reverse vaccinology, basically, you take the genome-based approach. You take the genome, compare the genome, make some subtractive genome approach. You find out the through the genome analysis, pan-genome approach analysis, to find out the very specific antigen which could be potentially used for as a vaccine candidate. Here, instead of targeting whole organism, you can pinpoint, you can basically use the very narrow range of antigen, and that antigen can be tested further for immune system immune activation. That's called antigen selection and validation. It takes usually 1 to 2 years. There are many success stories about reverse vaccinology we'll discuss in 1 or 2 slides, because I selected this topic to show some of this impact, and then later on I will share one of the thing which we have published recently. And then this era is basically is called structural vaccinology. Even if you identify a antigen could be the protein molecule, but not all the part of the protein is immunogenic. But we say the part of the protein which is immunogenic is called epitope. How do you get the epitope information? There are the many. If you talk about the 100 kilodalton protein, which is really immunogenic or which can trigger the immune response, but among this 100 kilodalton, probably you will have a few pocket of the protein which are basically is epitope, which has antigenic ability to activate the immune response. So how to get the information? Where exactly the epitope is present under the structure or under the protein? If you get the structure information and use this, define where exactly the epitope, whether the B cell or T cell. T cell could be basically the linear information, but mostly the B cell where structure vaccinology really plays an important role to define the area or define the region which on the protein, which is a potential epitope, which can be engineered further to make a many sort of multi-epitope vaccine design. This is a really important area. Now, when I say the structure vaccinology, let me explain what is the structure in fact. But before that, what do you mean by epitopes? There are different methods to identify epitope. One is, of course, in a normal, if you go any immunology lab, you'll find that they do ELISA, they do immunofluorescence, radio immunoassay. I don't want to explain all those things, but this is a normal way to identify epitopes present on an organism. But if you really want to find out what is the structure of that epitope, you need to go for any structural technique. So one of the structural techniques is called X-ray crystallography. There are two more techniques are there. Which I mention X-ray because I know some of my specialization is X-ray crystallography. The X-ray crystallography approach is basically where we take antigen and antibody complex. And this antigen antibody complex was crystallized and scan for the residue between the antigen and antibody and find the epitope, which are really important for making the complex with the antibody. Now, this is the X-ray crystallography. Now, it takes time because when you do a structure and it is really, we need to find out whether the particular protein is going to crystallize or not. But however, the third method, which is very commonly these days, is called production method. Because we know the PDB database, we have plenty of the structure information already available, and you use the production. You do use all the bioinformatics tool and identify those antigen antibody complex, and then use the amino acid sequences of your protein and identify the epitope which can be calculated. So these are basically three major methods for identifying epitopes. Now, let me, for the interest of students, those who are there as a participant, for them, let me explain what is the basically X-ray crystallography. X-ray is everyone knows the X-ray is electromagnetic radiation, and that what we do with the crystallography is basically a spectroscopic technique where we study the interaction of X-ray with the matter. Matter here is in a protein molecule, and it gives the image of molecule in 3 dimensions. When I say, many of you might have seen a textbook that the protein has a 3-dimensional structure. It has alpha helix, beta sheet. And basically, if you see the structure of a protein is defined in 4 stages: primary structure, tertiary, secondary tertiary structure, and quaternary structure. These structure information of tertiary and quaternary can be calculated or can be estimated through X-ray crystallography. It gives the atomic detail. Here is what I took. This technique is very specialized to give the atomic detail of molecule in 3 dimensions. How the atom, if you see, talk about the amino acid glycine, it has a carbon, hydrogen, and the nitrogen. How these carbon, hydrogen, nitrogen are oriented or placed in a coordinate of 3 dimensions that can be calculated or estimated through the X-ray crystallography. You know, the major contribution of X-ray crystallography in biochemistry is the Watson-Crick model of DNA. Everyone knows how DNA model was generated. through the X-ray crystallography, and then we came to know that how A pairs with the T or C pairs with the G, and what are the rotation, how many angstrom one helices, one helix takes. All those things we know where we came to know through the X-ray crystallography. And then many of the phenomena like industrial mechanism of protein DNA interaction was understood. Basically, this technique provides a major link between the structure and function of nucleus. Now, there are two other techniques which are called nuclear magnetic resonance and cryo-electron microscopy, which is a recent one. These two techniques also very important for determining the three-dimensional structure of macromolecule or protein. I'm not going to go to the detail of this one, but briefly for the interest of a student, when I say then X-ray is before going to the X-ray, just let me assume that you want to see an object. What you see, if your object is there in the front of light, you see the object. In dark you can't see. That means there's a light interacts with the object and it makes a through the retina. Through the lens of your eyes, it makes the image. But however, if you go for simply is also simple microscope also work the same principle. If you put the light in the microscope and then see the object is made as a magnified image which you can observe. But however, how do you obtain an image of a molecule? Image of a molecule is difficult. You cannot use a microscope. Because why? Because molecule size is very, very small. As previously, the previous slide that I showed, that one of the conditions that any object to be seen, the light has to interact. In order for an object to diffract light and visible under magnification, the wavelength of light must be no longer than object. One of the things is light has a wavelength in nature. Like it's a wavy in nature means that is a wavelength and amplitude. The wave of the light should not be longer than the object. If it's like in the case of whatever we see in a visible light, it has a 400 to 700 nanometer wavelength. Hence, but in our atom, when we talk about the molecule, in the molecule, one molecule is attached with the many atoms. We'll talk about the water molecule. Water molecule, we have a oxygen and 2 hydrogen. That means 3 atoms. The 3 atoms, if you see atoms are attached to the molecule with a distance of 1.5 angstrom apart. It's not more than that. But if you take the visible light, whose wavelength is 700 nanometer, cannot interact with this one. Hence, we need to find the inner electromagnetic radiation. Only the X-ray, which is very important, whose wavelength is near to the interatomic distances, and then hence you can use the X-ray for diffraction. The individual atom diffract X-ray is still. Now we have finalized X-ray can be used to diffract with the atoms or molecule, and then you capture the image, and then X-ray still the possibility is still you cannot able to solve the structure because you need to have focus the X-ray. X-ray focusing the lens is not possible. Hence, the single molecule is a second point, which is a constraint for even you finalize the X-ray. The single molecule which is in solution, it cannot be is very, very weak as scatter of X-ray. Hence, you cannot capture it. But however, if you see before to get the structure, we need to have a protein or molecule should be arranged in a crystal form. Hence, in a crystallography, one of the important steps for crystallography is called getting the crystal of a molecule. And then you take the crystal is formed, and then you use the X-ray, and X-ray will bombard onto the crystal. It diffract the light, and light is captured where the intensity and direction is calculated, and what you see is electron density map. On this electron density map, you model the structure of protein molecule. So this is basically what we do in our study. But this is very popular technique which have been used to solve the many structure of a protein. If you go to PDB database, you have plenty of structures whose three-dimensional structure are solved by X-ray crystallography. Now come to the vaccinology, structure vaccinology approach. The approach is in the X-ray vaccinology that you need to get the pure protein or pure antigen from the pathogen, and that antigen is used to determine what you design. Once you have antigen, you determine the epitopes or map the neutralizing epitopes, discover the neutralization. Then you identify the domain which is responsible for neutralization, stabilize the antigen by the conformation. This is called a structure-based engineering. You make many epitopes together, fuse together, and then once all those all those things is ready, like you can select the best epitope, engineer it, and make many epitopes together, and that is that yeah, that rational manipulation of immunodominance can be done through the structure-based approach. And then what you do, the final rational formulation can be made through the multiple epitopes, and then the next generation of vaccine that you can use this one along with the adjuvant and then subject for further testing. So these are basic steps in structure vaccinology. Now, one of the few, since I made the topic as the impact, I will give some example where this is successfully done. One of the protein from meningitis, Neisseria meningitis, serogroup B, has a factor H binding protein. The factor H binding protein has a 3 variant. Variant 1, variant 2, variant 3. 3 structures were solved separately, and they have identified the epitopes, and then those epitopes from each variant was taken out and then grafted into the variant 1. And then what they generated is called engineered antigen, and this engineered is found to be very, very potent vaccine against Neisseria meningitis. Another example is like how the beauty of structure vaccinology is I'm going to give here. Here, one of the antigen-based domain was from Streptococcus aureus, where they found a protein called what's it called, fusion protein. The protein is which is called pilus protein. The pilus protein BP2A has they identify their domain, which are responsible for basically which has epitope, and those, if you see, BP2A is a 3-domain protein, and we don't require all 3 domain for all 3 domains are not required for antigenic immunity. What they found, only a D3 domain was important for activating immune system. They used the D3, this is structure information, and then plenty of D3 domain, similar domain was engineered, and then they were linked to the linker, which is GSGS, glycine serine linker, and they made a chimeric antigen. And this chimeric antigen is now very, very popular, and they found to be very, very immunogenic. And similar approach was done for respiratory viruses, and engineered in one of the antigen of influenza virus also. So these are 2 examples which I have mentioned here. I already mentioned about the reverse vaccinology. Reverse vaccinology is the process where you can just find out the genome of non-human and pathogen with the protein which is not present in human and which are only exclusively present in already. Hello. Those reverse vaccinology approach can be done where we have used all the bioinformatics tool. We say that there is a very important for substructive proteomics, which involves is very cost effective. That instead of screening all thousands of protein, you can able to screen only few proteins for its vaccine potency. So basically, this is immunoinformatics technique. What you can say also we say as a bioinformatic approach of immunology is called reverse vaccinology, which involves analysis of complete information of organ. Now, one of the worldwide effort is a COVID-19 vaccine disease, like plenty of info. If you see COVID-19, throughout the world, every lab who's specialized for bioinformatics or structure biology or immunology, they are using all different approaches, either reverse vaccinology, structural vaccinology, and then immunology. Altogether, they found one of the protein which is called a spike protein, very potent. You can see the bottom panel, which has a structure of a spike protein, is already solved in recently, and it was found to the 4 domain, N-terminal domain, receptor binding domain, and you have a C-terminal domain, is a green color, and this one monomer, the 3 monomer combined together to form a spike protein. Then if you talk about these efforts are going on to develop the vaccine. Right now, the 3 vaccines which are available based on mRNA, another is wearable on the viral vector we use for to express the spike protein, and the protein itself was one of the different approach was going on along with the adjuvant. So that adjuvant and protein can be injected, and then you see the effect of efforts made using reverse vaccinology and structure biology for vaccine design for COVID-19. Now come to my area of research. Currently, in my lab, we are focusing three-dimensional structure of determination of virulent factor and outer surface protein from different pathogenic organisms. Why we are doing that? We are doing to understand the basic study the interaction of these protein with the host protein at atomic level, and we use primarily X-ray crystallography analyze the function of these protein through the biochemical and biophysical technique. We also use the structure vaccinology bioinformatics approach to design the potent vaccine. So with this, the model organisms we have chosen in our lab is three organisms: Mycobacterium tuberculosis, pathogenic Leptospira, and Helicobacter pylori, which is the focus of my lab. Today I will only present about the Mycobacterium tuberculosis. As you know, tuberculosis, everyone knows, is an infectious disease caused by Mycobacterium tuberculosis, which infects the lung. And then if you see that tuberculosis are basically categorized in the 2 types. One is active tuberculosis, another is the latent tuberculosis. Active tuberculosis is called active pulmonary tuberculosis, where people really have symptoms. And latent tuberculosis is many of us have many in India also will show the data. Most of the people, like you can say that 10% of people are only having active tuberculosis, 90% have a latent tuberculosis. So what do you mean by latent tuberculosis? Latent tuberculosis is basically where the bacteria sits in the macrophages of the lung in without any action. And the moment you have your immune system is compromised, that latent infection can be relapsed, and then it can reinfect your comes in the active infection. So now, most of the time you see that we have a BCG vaccine which is available, which is effective up to the childhood or not more than 5-year age. For adults, we don't have any vaccine. There's still, and then the vaccine against the latent tuberculosis is not available. So these are challenges still people are facing, that we don't have a vaccine available for the adult, or we don't have a vaccine for the latent tuberculosis. Now, in the same time, they have a multi-drug resistance is also coming up. The extensive drug and multi-drug resistance makes the TB or is very important organism for which infect human. Now, if you talk about the total genome, 60 total infection. 67% is like global infection is active, 33% lately infected. This is a global estimate of TB. This shows that if you see the country, this shows green color shows different shade of area shows the infection rate or incident per 1 lakh. If you see India comes under the 100 to 199, 100 to 200 incident per 1 lakh is also is there in India. It is a recent World Health Report. But this over the period of time, if you compare with the last 1 decade, we have made a lot of contribution to control the TB epidemics in India or globally also. But still, this is still not completely eradicated because we don't have vaccine. Though we have a treatment that we can use the different stages of antibiotics. Which can go up to 6 month to 9 month period, but it's a longer duration of antibiotics. Treatment with antibiotics makes even bacteria for multi-drug resistance. So these are the challenges. With these challenges, my focus was dormant stage. Many of this we have bacteria or MTB resides in the macrophages as a dormant stage. When I say dormant, means without any metabolic activity, without any any active replication. It sits into the macrophages ideal in a latent form without doing any work, anything. But because a lot of microarray data shows that the 50 genes which are upregulated in dormancy under the transcription factor called DOSA, it was found that 50 genes or almost 50, approximately 60 genes are upregulated in dormant stage of bacteria. When bacteria sits into the macrophages without any active infection, where the whole idea is to really eradicate or to remove bacteria even from the latent infection also. But microarray data suggests that there are 50 genes which are quite important, and these 50 genes are called dormancy survival regulon. That means DOSR regulon. The DOSR regulon are basically a set of genes which are under the control of dormancy survival regulon. And it says that most of the function of this DOSR protein is not known. However, many of them were found to be the good T cell antigen. These are the report which I am telling that people have already contributed. DOSR regulon upregulated in latency in MTV as evidence from the microanalysis. Certain DOSR regulon proteins are really hypothetical and whose function is still not known, but they perform really they're immunogenic. Among the DOSR regulon, if you see that are plenty which I have listed here, don't worry about the protein name. In MTV or mycobacterium tuberculosis, we name particular protein like RV2626C, 2032. These are the antigens. Here, these are the list of few proteins which are really involved for one or other way into the T cell activation. Some are involved for novel macrophages induction. Some are involved for mediating TLR2 mediated pro-inflammatory cytokine secretion. Some are reported to potential IF and gamma induced response. These, and then moreover, there's RV2032, RV3127, RV3131 are also included among the list of 45 antigen an attempt to design a vaccine against MTB. So these are the background information of DOSR, importance of DOSR regulon, or importance of protein which are under the DOSR regulon are important for inducing T cell and B cell response. Among this list set of 50 genes, what we found, there's a class of protein called nitroreductases. When I say nitroreductases are the enzymes that reduce a nitro compound into their hydroxyl amine and amino form. And these nitroreductases are basically two types: is oxygen insensitive nitroreductase and oxygen sensitive nitroreductase. Oxygen insensitive, they reduce directly by transferring 2 electron to the nitro group. Oxygen or type 2 is oxygen sensitive, they reduce indirectly by the transferring the first electron to the forming the nitro anion radical. These class of enzymes, which is called nitroreductase, also found to be very, very immunogenic also. What in my work, what we have taken the putative nitroreductases from DOSR regulon, and because those nitroreductases, which is overexpressed, whose expression was found to be quite high compared to the normal bacteria, and these were even tested in the previous study, showing there's some other way their involvement into the activation of immune system. So we have taken 4 putative nitroreductases from DOSR regulon and use this immunoinformatic techniques to design a potential chimeric vaccine. Instead of now, if you really want to test, you can all the 4 protein you purify and then go for determining the epitope. But what the beauty of using bioinformatics or immunoinformatics, that you take this 3 protein or 4 protein, which I have targeted a 4 protein from nitroreductase class of protein, and use this immunoinformatics or bioinformatics techniques, even if you use the structure-specific circumnumeric power, because the structure is still trying to solve the structure of this protein, but success is not there. However, the immunoinformatic techniques we have used is if you see the workflow which follows, like you can take all the 4 protein which I have targeted. I will mention the name of these 4 protein, and then use the bioinformatics tool to identify the MHC Class-1 and a Class-2 epitope. And then among this Class-1 and Class-2 epitope, you find out the promiscuous epitope. You select the epitope which are really promiscuous. You select the epitope which are non-self or antigenic. Yeah, and then we selected the IFN IL4 and IFN gamma induced for MHC Class-2, and then you use those peptides which are really important epitopes and we talked on into the HLA allele, see this stability or interaction with the, and then we have gone for design a subunit vaccine, or we design a chimeric vaccine, and those chimeric vaccine are further tested for their indigenicity, physiochemical properties, secondary structure, solubility, and other thing, and then we predict the three-dimensional structure. And what we see here is I'm going to give the result of which we have done it. Now, if you take the example of we have selected the four protein RB, four putative nitroreductase. One is RB2032, RB3127, RB3129, RB3131. So these four are known to be nitroreductases, involve have a high level of expression in the microarray data, and known to be a potent vaccine candidate. But our idea is to get the epitopes from these four protein and generate a chimeric vaccines, where only you select the epitopes from which are potential ability to activate the immune system. So since I have used all bioinformatics tool, here this graph shows the binding profile of T cell epitopes with a globally present HLA. You know, HLA is an analog of MHC Class-1, analogous to the MHC class MHC in animal. HLA is a human part which presents the which presents the epitopes to the T cell. Now, if you say there are 36 HLA A type, 34 HLA B type, or 10 HLA C allele present throughout globally. What we have selected, we have estimated the predicted the peptide which shows the maximum binding on this allele. In the first panel showed for MHC Class-1, and then second panel showed MHC Class-2. Or on the x-axis shows different label as a different HLA, and even similarly, and the y-axis shows the number of peptides. This shows that the peptides which are overlapping peptides which are generated from all the four protein. Color codes show the different protein, so all four proteins, and based on that we get the information that out of if you consider MHC Class-1 from RB2032 and RB3127, these are total number of epitopes generated which can bind to different HLA globally, HLA allele. And now epitope which is binding to one allele, we have selected the epitope which are binding more than 3 allele, like 51. So these are the basically table shows that that what are how many numbers we have selected, and then you made a further screening based on the promiscuity. If peptide bind more than 3 allele, we say the promiscuous epitope. Then we went for further testing those predicting the indigenicity. We found that out of 15, 19 was showing the more number of antigenic epitope in case of 2032, respectively for other also. Then we go for self and non-self epitope, because many of the epitopes could be having similar epitope present in a human body. Then we always check there should be non-self. So self will be discarded, or non-self can be considered. Similarly, we have done for MHC Class-2. MHC Class-2, if you see here, in MHC Class-2, we have put two additional steps. We have a screen for IL-4 inducing epitope and IFN gamma inducing it. So these are two cytokines basically involved for in case of bacteria infection, in case of MTB infection. So these are cytokines which trigger, activate the macrophages, which activate the T cell, or even the production of MHC Class-1 molecule. Then if you see here, finally, the number has screened, number have narrowed down, and to 6, 3, 2, 7, something like that. Now, the final which have MHC-1 and MHC-2 epitopes and their position on the protein, if you select, if you say what we found MHC Class-1, that these are the epitopes which are present, and whose score are Vaccinology score means the score which bioinformatics tool we have used, which shows the approximately 0.9 and more than 0.9 score onto the antigenic score, and its positions onto the protein are also listed. Similar the case for MHC Class-2. Now these are the final epitope we have considered. Now among the final epitopes, we have taken one epitope which are from each representative protein to see their ability to bind to the HLA allele. Now, how do you see the binding of HLA allele? We have a HLA allele structure available, three-dimensional structure. Those peptides were allowed to bind onto the, to dock onto the structure of HLA allele, and then we see the we have done the dynamics, pharmacodynamic simulation, to see whether the stability of these peptides are really present, is not being thrown out from the cavity. And then we calculate the number of interaction on the C panel, show the how many number of hydrogen bond are involved for making stabilization of this peptide into the HLA class 1 HLA allele. Similarly, one of the allele was considered for MHC class 2 epitope, and there also we have done the similar thing. We have done the molecular dynamic simulation to see the stability into the group, and we have calculated the number of interaction and hydrogen bond information. But basically, these two approach clearly shows that the peptide we selected is really having stable interaction with the HLA allele under the structure. Now, if you finally, after this analyzing the peptide which is really showing the stable interaction with the HLA, we have selected those peptides, and from each protein, if you see a protein color is labeled into the blue, red, green, and orange, and the peptides are taken, and those peptides are connected together by the linker. Here linker is, we have made alternate linker, like AY means alanine, and tyrosine linker. We have also used glycine, proline, glycine, proline, glycine linker to link these peptide. And then after that, we use one of the adjuvant. Here in this case, we have used one of the adjuvant which is really important for TLR2 agonist, which is phenol soluble modeling alpha 4. This adjuvant was used, and we constructed a vaccine, and this vaccine construct was further predicted to be a physical and chemical properties. We found the molecular weight of this construct is 26.5 kilodalton, have a theoretical PI is 10.1, and stability, other thing we have checked it. Okay, now this vaccine contains the epitopes from the 4 different proteins, and they are found to be quite predicted to be quite stable, and quite half-life is also stable. And then we check their secondary structure, and we found the secondary structure of this vaccine construct was 49% alpha helix. The majorly more is the protein has alpha helical structure, and 9.6% of beta sheet. And then these are related to the random coil attached to these helices are attached to the random coil. So this is a basic information about the construct which made through taking the consideration of different epitopes from 4 different proteins. Now we use this construct to estimate what is the tertiary structure of the multi-epitope vaccine construct. We found that is, as secondary structure shows the mostly alpha helical structure. Here it shows alpha helical structure, and then any structure where you do predict or you make the tertiary structure, we need to see whether it's validated the ideal conformation or not. For idealization, we check the Ramachandran plot. In the Ramachandran plot, it shows that most of the residues which are modeled onto the tertiary structure of the protein is present onto the allowed region. The other is the prosa value is also this black color dot lying onto these values, Z score values, which clearly shows that the structure which you have modeled is really ideal structure. Now, next step is if we test this protein. If any vaccine, if you talk about vaccine molecule which induces immune system through the TLR mediated or TLR4 or TLR2 mediated activation of cytokines. In case of TB, TLR2 is more important. Hence, we have taken the TLR2 structure, and we use our structure. The blue color background, blue color structure is a TLR2 structure, and green color is our vaccine construct. We have docked it, and after docking, we analyze the interacting residues, which shows that the vaccine construct is talking perfectly fine with the TLR2. However, this is not the final estimation. We need to go for molecular dynamic simulation to show that the dog structure is really stable onto the TLR2. We have gone to the molecular dynamic simulation, and if you see here RMSD, which on an average RMSD is 0.6, which shows that it hasn't come out from this complex. The complex is fairly stable. We have analyzed the root mean square fluctuation of residues. This always shows a fairly stable. We have analyzed the hydrogen bond, which shows around on an average 12 hydrogen bond formation to make this complex stable. If you see the radius of gyration values, which also indicates about the compactness of these two complexes. The compactness of two structures also indicated by the radius of gyration shows that the vaccine construct is really stable in complex vertical form. The last, which have what we have done, we have used this construct to predict this induced immune response through using a bioinformatics tool. What we found that in the first panel it shows that IF and gamma is slightly high. Blue color panel shows slightly high apoptosis. If you assume that this is a vaccine construct, if you really want to inject to activate the immune response, you always measure the IF and gamma. Here IF and gamma is significantly high. And in case of B and C panel shows that TH response is high, TH1 response is high, which is quite good for in case of MTV infection. And then lastly, it shows that TH1 response is predicted to be quite higher in case if you assume that this vaccine construct is injected into to induce any immune system. Now, this data suggests that whatever the vaccine construct we have generated could generate an effective immune response. And then this is all about what we have recently we have published this also. In conclusion, which I say that which I highlighted the importance of the structure vaccinology and bioinformatics, giving some examples where the success story of vaccines where the structure and bioinformatics tool was involved. And our study uses a immunoinformative approach to evaluate the immunodominant promiscuous epitope from putative nitroreductases from dorsal. And we have selected the non-self epitope and fused together with a linker and adjuvant to construct the multi-epitope chimeric vaccine. And these vaccine constructs found to have stable interaction with TLR2, and stability was analyzed through molecular dynamic simulation. And this construct may give good immunogenic response. However, this all are in silico work. In vitro and in vivo validation of this vaccine construct and the protection against infection are really has to be tested. So this in vitro in vivo validation of this vaccine construct is still under progress. This is all about, and then any work is without acknowledging the people behind that is no use. I really want to acknowledge for my group, which is there are 4 PhDs who are working here, and then I presented the work of Shiraj and Surabhi Lata, who have contributed to for this work. And then now I really want to acknowledge my collaborator in IAB Hyderabad, and then Professor Chang from Cornell University. And I'd like to thank my for funding from DBT and DST or SERB, and then University of Hyderabad Institute of Eminence funding. These 3 funding made our lab to establish in University of Hyderabad. And finally, I would like to thank all of you for patiently listening my talk, and I'm ready to take any questions. Thank you very much.
Thank you, sir, for your wonderful words. It was very informative and it means a lot to us. And now it is an interactive section, and our audience can raise their doubts if you have any.
Looks like there's no question. Either everyone has understood or nobody has understood anything. Sir, how? And there's one question. Okay. This not about the protein inhibition. Anjana asked like how did you select the ligand from thousands of ligand available in repeated data? First, let me explain to you, Anjana. This not I haven't selected any inhibitor. What I have used this peptide. The peptide we have selected. Those peptides were docked under the HLA allele and see their interaction. It's not about the inhibition. Hope is understood. Any other question?
Good evening, sir.
Yes.
Sir, how to identify epitope?
Bit of?
How to identify an epitope?
Yeah, I told you that if you see that what I mentioned, the importance of bioinformatics tools available. So there's a plenty of bioinformatics tools available, either a free form or paid form, which we use to analyze those epitopes. If we, in our case, we have selected 4 protein. The sequence of protein was given to the program, and programs use some artificial intelligence or neural network approach to find out the epitope which can have a potential to bind to the HLA, and that program gives the output in form of different peptides. Those peptides you select for as epitope.
Thank you, sir.
Do pH inside the cell determine the stability of vaccine? Yes, of course. The pH can determine the stability, of course, but we haven't considered the pH. But whenever you do the simulation, we use the physiological pH. When we set up our protein interaction or protein or ligand interaction, what we set up, or we generate a molecule or a protein molecule such a way that you provide a similar environment or as a physiological environment. We provide a salt, we provide ions, we provide pH, and that you select as if you are simulating really the physiological condition in your in silico study. And there is, what is the genome size required for mRNA and DNA vaccine development? For mRNA and DNA, there's no. Yeah, for mRNA and DNA vaccine, we don't require a big genome. We need to identify, first thing you need to identify the potential potential candidate. Like in case of coronavirus, if you know that we have the study has already done and they have identified the spike proteins are really immunogenic. In case of mRNA, they have used mRNA based vaccine. They have used basically mRNA of spike protein, and that spike protein are basically engineered such a way that can you can make a mRNA of that spike protein, which can, when it's injected, it can express the protein. Is no, it doesn't mean any genome size. It doesn't have any dependent on the genome size for making any mRNA and DNA vaccine. We need to select, for if you're talking about mRNA and DNA, you need to select which is the potential target. That's okay. Second question, third question: PDB database structure derived by NMR X-ray crystallography is available for docking. Which structure is suitable? One is derived for NMR. Yeah. So basically, you can use both structures suitable for a docking, no problem. But however, NMR has ensembles of a structure. NMR, if you find out the NMR coordinate have a many ensembles of a structure. Like if one NMR structure will have 12 PDBs. Hence, I can answer that you can use either NMR and X-ray, any of this structure. However, you make sure that if you're selecting NMR for structure for docking, you make sure that you select only one ensemble, not all. Next question: is there any criteria to select the linker as EAK linker? What is the significance? So this linker and all, there are many report people have done the wet lab, or basically the experimental work, which shows that which linker could be feasible for your thing. So there's no specific rule. As long as your protein is stable, your protein is can be folded accurately or in a 3-dimensional, you can try different linkers. Any linkers which can make your protein to be folded can be used. For our study which we have selected, we have tried many combination, and we checked which combination gives better folding response of our protein of interest. Any other question? Yeah, significance of linker is, of course, significance of linker means that it can provide the really proper folding of your protein. Since you have taken the region from different protein, and those region you are trying to merge and combine together to make a single protein, so those region has to be linked to one another by some sort of linker, as long as this linker will not interfere the folding of the protein. If your protein is not folded correctly or accurately, you may not able to go ahead for purification for generation of protein in vitro. Any other question?
Sir, what is the criteria for naming of a protein? Example RV2032.
What is the criteria for naming RV?
Yeah.
There's no criteria. This RV is somebody has done. RV is, even I forgot. RV has some name for that. RV is when the whole genome sequence of Mycobacterium tuberculosis was done in 1998, and after this genome sequences, the annotation of gene was done, and in that annotation they have put this name of each gene, and that is RV a number. RV stand for really has gone out from my mind. RV stand for some name. RV you will not find anywhere else. RV will find only the Mycobacterium tuberculosis. I'm sorry, really, I forgot what is the RV means.
It's okay, sir. Thank you, sir.
Yeah. Any other?
Anjali?
Sir?
Yeah, move to next session.
Okay, ma'am. We are about to the end of the program. Expressing our gratitude to all of you is also one of our priority. For that, I invite our convener, Dr. Preeta Pillai, for vote of thanks.
Thank you, Anjali. A very good evening to everyone. It's been really pleasure being with you all today with the concluding vote of thanks remark. Myself, Dr. Preeta Pillai, Assistant Professor, Department of Botany, SVR and SS College, Vazhoor, would feel great privilege on behalf of SVR and SS College, Vazhoor, and Zoology Department, Botany Department, and IQAC to propose vote of thanks on this second day of our webinar series. First, I would like to start my in-deep gratitude and heartfelt thanks to Almighty for making this webinar series a resounding success. Together, would like to show my gratitude to my platform providers, my college, Sri Vidyadhiraja NSS College, which is affiliated to Mahatma Gandhi University, blessed by the Chatambi Swami Tiruvadikal and Bharat Keshri Sri Mannatha Padmanabhan, run by the Nair Service Society. Let me come to my main duty of today's session. I admire one of my leader. She is not a celebrity, not much popular person, but she is a leader among us. So with full support and zeal, she is our principal. She is Dr. Renuka MR. I really owe my gratitude to her. Thank you, ma'am. Thank you for your ever supporting nature with us. Now, I would like to express my gratitude to our second day webinar delegate, Dr. Muhammad Akif, with the topic, the impact of structural vaccinology and bioinformatics on vaccine design and development from School of Life Science, Hyderabad University. Sir, let me explain what all things briefly about what you all have explained today. That you have started with the simple topic like what is vaccine, the immune system and all. Later, you have come to your advanced research work, like the adaptive immune response under this two major arms you have explained, antigen presentation and recognition, how to do. Very much fascinated and seeing by structural vaccinology technique and all. It gives deep insight of methods to identify epitopes. In this, you explain X-ray crystallography, other experiment techniques, images, microscopic objects and all. You made us enlighten about how we can obtain an image of a molecule. We really liked to listen you approaches for such structural vaccinology. Two different examples also you have shown us. You have explained the reverse vaccinology also. After this COVID-19, we heard many times about the spike protein term. So that also you tried to elaborate us. You also elaborated the DOS-R regulon protein, number of predictions, how to predict epitopes and all. Sir, these all were very advanced work which you have shown us. So we could conclude that you are doing such a wonderful thing in the area of research. So we were really fortunate to have you as a resource person. Thank you so much, sir. As it was a series of webinar with 2 days presentation, hope many of them attended yesterday's presentation as well. Would like to brief about yesterday's presentation bit. It was presented by Dr. Irfan Ahmed Ghazi, Plant Science Department, University of Hyderabad, with the topic Functional Genomic Approaches for Plant Disease Resistance, Rice an Example. So he also explained about the topic genomics, whole genome sequencing, structural genomics and functional genomics, and importance of functional genomic approaches for plant disease resistance. He also made us aware about the software available for gene predictions and protein database. Once again, I would like to thank both of you, Dr. Irfan sir and Dr. Akif sir, for taking out your valuable time with us and enlighten us with the latest innovative research work. Thank you, sir. Thank you so much.
Thank you very much. Thank you for your night.
My gratitude is void without saying thanks to all my colleagues, Dr. Jay Kumar K, Dr. Rajesh MG, Mr. Babin B, Dr. Prashant Kumar S, Mr. Gokul. They all worked hard behind the curtain to make this webinar series resounding success. Thank you all. Last, not the least, my special thanks to my dear teachers. Most of the teachers, those who are attending here, they all are my friends as well. And together, research scholars, PG, UG students, and dear participants, you all were a great listeners. You all made this webinar series a great success. Malavika and Anjali, you did your job very well. Thank you both. Thank you once again. Thank you everyone for making this